science lovers! Today we're going to talk about how to distinguish from a bread made with sourdough and a bread made with baker's yeast. For those of you who haven't heard of sourdough before, it's a traditional fermentation method that uses a natural mix of lactic acid bacteria and yeast to make the bread rise, instead of using, for example, baker's yeast that can be bought at any supermarket. This natural mix of microorganisms comes basically from mixing flour and water and letting this mixture contaminate itself with microorganisms from the environment. So, for example, you can get the bacteria and the yeast from the flour, or from the water, or even from the air. There are many benefits from using sourdough to make bread. For example, the bread has a nicer taste, a nicer texture, and it lasts longer. All of these reasons have made sourdough fermentation more and more popular over the years. And now, between 30 and 50% of all the breads made in Europe are made with sourdough fermentation. The problem is that up to date, there's no standard way of authentication of which breads are actually made with sourdough and which ones aren't. The only form of authentication is what the manufacturer says and how the bread tastes. And this is a problem if we want to be sure that what we're buying and what we're paying more money for is authentic sourdough fermented bread, or whether it's just bread made with baker's yeast. These researchers have developed a way to measure directly in the bread the amount of lactic acid bacteria. And this can be used as a standard way of saying whether the bread comes from sourdough fermentation, in which case it would have a high amount of lactic acid bacteria DNA, or whether it comes from using baker's yeast for the fermentation, in which case the amount of lactic acid bacteria would be very low. These researchers have developed this method by using qPCR, or quantitative PCR. This is a method that can be used to quantify the amount of specific DNA in a sample. And it's a method that is widely used in food sciences to detect specific ingredients like allergens, or to quantify microbial populations in food for safety reasons. It's also interesting to note that when DNA is subjected to high temperatures, it tends to break. So, for example, if you want to study the amount of bacterial DNA in bread, but DNA breaks when you cook the bread, then it could be possible that you won't detect any DNA of the species you're looking for. To avoid this problem, what the researchers did was, instead of trying to study a large amount of DNA, which would probably break in the cooking process, they chose to study a very small portion of the DNA that would be big enough to identify the bacteria it belonged to, but small enough to avoid it breaking during the cooking process. So I want you to keep in mind that sourdough fermentation is a popular baking method that is used more and more every day, and that needs some form of standard authentication, like the one that these researchers proposed in the paper, in order to avoid fraud in these types of products. Thank you very much for watching. I hope you enjoyed the video, and if you have, you can give me a thumbs up and subscribe to my channel. And as always, I have left the link to the paper in the description. See you next week.